Hello, my name is Max Distler, and I am a PhD candidate in the Department of Chemistry at Northwestern University, and I earned my bachelor's degrees in chemistry and neuroscience from Wesleyan University. And today I would like to talk to you about precision nanomedicine and the role that nanotechnology is playing to expand that field. But in order to fully grasp the potential of this field, we have to start at the very beginning, with a discovery that changed our understanding of biology forever. About 70 years ago, James Watson, Francis Crick, and Rosalind Franklin defined the structure of DNA and shared it with the world. This discovery empowered further research, which led to the ability to synthesize DNA, understand how it works in relation to disease, and develop new medicines based on it, completely altering the way in which we can attack and treat diseases. So, precision nanomedicine, or at least the brand that I'll be discussing today, does not rely on small molecules as therapeutic agents, but rather it uses short snippets of DNA and RNA as medicines to flip genetic switches and correct or even eliminate disease. But this is just one piece of the puzzle. It turns out that in order to develop medicines broadly, you have to be able to get those medicines to the organs and tissues that matter. But when we systemically inject DNA into people, it very rarely reaches the point of interest in the body, and it's often broken down far too quickly to have any sort of therapeutic effect. This is where nanotechnology comes into play. About 30 years ago, our lab developed a new synthetic form of DNA and RNA, where we can take the same exact short snippets of DNA, but arrange them in a spherical architecture around a nanoparticle core. These spherical nucleic acids, or SNAs, have vastly different properties from the naturally found DNA that Watson, Crick, and Franklin identified all those years ago. And it's these differences that make them such a promising platform for developing new precision nanomedicines. For example, any cell that comes into contact with the spherical nucleic acids takes them up to an extremely large extent. And we can use this capability to 1. Expand the field of therapeutic development, and 2. Develop new medicines to treat diseases that conventional medicines cannot. For example, we can incorporate the SNA into creams and deliver them topically to treat skin conditions. We can put them into droplets and deliver them to the eye to treat diseases of the eye. And we can even use them to target the brain in order to treat specific types of brain cancer. Now, most recently, we put the SNA to the test. We ran a first-of-its-kind clinical trial in which we used the SNA to treat one of the most aggressive forms of brain cancer out there, glioblastoma. And the results were really quite remarkable. First of all, we found that the SNA had no toxic or adverse effects on the patients. Second, we saw that the SNA was able to penetrate into the brain, and even more importantly, into the tumor cells within the brain. And third, and most impactful of all, we found that as a direct result of SNA treatment, the intended target was being eliminated. Now, this is just the first step down an extremely promising path towards developing new, powerful precision nanomedicines using the SNA platform. And all of this work has been made possible because of the support of dedicated and interested donors. Specifically for the glioblastoma work, Ron and JoAnne Willens played a huge role in making it happen. And we are always so grateful and appreciative for the support that the Willens family and many, many others have provided. As for the work inside the laboratory, all of that is possible because of the collaborative environment that is nurtured at the IIN and at Northwestern at large. For example, I'm a chemist, but every day I get to work with physicists, engineers, and medical doctors to develop new types of medicines to treat innumerable diseases. Thank you.